Okay. So first of all, thank you for this introduction and also for allowing us the opportunity to speak in front of you today. So I'm really excited to present my work in front of you. So for today's presentation, I'm going to talk about my Master 2 internship that I had the opportunity to do at the Université de Lausanne. So my question was about the revision of the phylogeny of radiodonts using whole body characters. So first of all, I would like to introduce to you about the context that led to this question and starting with a presentation of the group, because I'm aware that Radiodonta is not really a common name. So Radiodonta is an extinct group of arthropods that lived during the Paleozoic, and more precisely between the Middle Cambrian until supposedly the beginning of the Devonian. So one of the most famous representatives of the group is Anomalocaris canadensis. Maybe you've already heard about it. They are really well known thanks to the structures located at the head that we call frontal appendages. So these organs, raptorial organs, help them carrying food into the mouth. But Radiodont is more diverse in terms of species and in terms of morphologies than just Anomalocaris. So I summarize on this slide the different kind of morphologies you can find within the group. So you can see that the shape changes between all the different organs of these animals, starting with the frontal appendages. So different shape reflecting different ecology. There's also the head. You can see a structure that looks like a shield. It varies in shape and in size. You also have the tail, if you have one or not, what's the shape of the tail, the number of flaps. So yeah, this is a pretty much diverse group. However, our understanding of the group is still poorly known because there is some bias in the fossil record. In fact, there is only one structure that can be easily found, and I'm talking about frontal appendages. So frontal appendages are the sole structure which is sclerotized. So that means that it can be preserved more easily in the deposits, while the rest of the body decays and is then much harder to find. So because of that, a lot of studies focus on the study of the frontal appendages, including phylogenies. So here on this slide, you have one of the latest phylogenies published when I was doing my internship. So as you can see inside Radiodonta, there is 4 different families, namely Anomalocarididae, Amplectobelridae, Tamisiocaridinae, and Herdidae. So these 4 families were defined thanks to the shape of the frontal appendages. But according to what I just said to you, there is now debate among specialists in order to see if we should continue using this organ or not. For example, some authors consider that this organ carries a lot of homoplasy, so we should be careful with the use of this structure because maybe the diversity is more a reflect of the ecology of the animal more than common evolutionary history. And on the other hand, there is other researchers that consider that it's a good taxonomic proxy because in the end, this is the main organ we can find, so we should study it. It's better that than nothing. So this is in this context that I'm asking the following question: are frontal appendages a good phylogenetic proxy? So this was my main work during my internship. So in order to answer this question, I put my work on data matrix. And I took a previously published matrix, so the one I showed to you in introduction, but I did some changes in this data matrix. So the idea was to reduce the number of characters coding for the frontal appendages because it's a well-known structure, so there is so many characters for them. So I tried to remove the characters coding for homoplasy and link to taphonomy. And I try to add as much as possible characters coding for the rest of the body in order to have like more stable signal. So once my matrix was completed, I analyzed it under parsimony analysis on the POP software, and I did 2 different kinds of analysis. The first one that I call the global includes all the characters and taxa possible, so 51 characters and 29 taxa for this matrix. And I did another analysis that I call the body character. So this analysis don't take into account all the characters coding for the frontal appendages, and I removed as well the taxa that were known only by frontal appendages. So in the end, I had 39 characters and 22 taxa. So now let's jump to the results and see what does the tree look like. So we start with the global analysis, and what you can see on the slide is the majority rule consensus tree. So you can see that radiodonts here appears monophyletic, and inside this group there is 3 different families, namely Herdidae, Amplectobellidae, and Tamisocarididae. So we have no sign of Anomalocarididae here. There is also another group that I would like to highlight, which is called the Deuteropoda. So this group is considered as a crown group arthropods. So the crown group in arthropods include trilobites, insects, crustaceans, spiders, etc. So normally the Deuteropods are fairly different from radiodonts, which are considered as a stem group arthropods. But in this analysis, they are found in a monophyly inside the group. Okay, so now if we jump to the body analysis, so here again, you have the majority rule consensus tree. You still find Radiodonta monophyletic, but this time only 2 families are found, Herdidae and Anomalocarididae. So no sign of Amplectobellidae and Tamisocarididae anymore. And Deuteropoda are still present, but in a paraphyletic relationship with other radiodonts. So now if we discuss further these results. So on the left, you have the global analysis, and on the right, it's the body characters only. So we can see that the trees topologies are completely different from each other. Only 1 family remains stable between both of the analyses, which are the Herdidae, and all of the others change depending on the choice of the focus we made on the body. Tamisocarididae, for example, it was previsible that it won't be visible on the second analysis because it's a group only known by frontal appendages. So, of course, if we remove the information, we remove the group. But for Amplectobellidae and Anomalocarididae, there are some differences. And there is also another thing that I would like to highlight, which is about the position of the Deuteropoda that questions the monophyly of radiodonts. So I will develop a bit this particular topology. So here you have in green a deuteropod, and one of the characteristics are on the eyes, for example. So the eyes of the Deuteropoda are sessile, so they are glued to the head. And on the right, you can see a radiodont with stalked eyes. So normally it's a characteristic of radiodonts to have stalked eyes and a synapomorphy of the deuteropods to have sessile eyes, but recent discoveries in 2020 found out some radiodonts with sessile eyes. So here we have a derived feature into the stem group that makes us suggesting that there is a mosaic of character composing this group. So here I use only 1 character to describe the point, but there was many of them coding in the matrix. So the more we discover with radiodonts, the less clear it is with the relationship with the other taxa. Okay, so now if we come back to the main analysis and compare it with the previous results. So on the left, this is my tree. On the right, it's the one I introduced to you. You can see that there is some differences. So first, in my analysis, I don't find a monophyletic Anomalocarididae anymore. And the relationships between the taxa change. So in the previously published matrix, Tamisocarididae appear sister taxa to Herdidae, but it's not my case in my analysis. I found them much close to Amplectobellidae than Herdidae. Okay, so that's important. Just before we go to the conclusion, I just would like to highlight some limits with my study. So first of all, the Bremer value. So all of the nodes are about 1. So we should be careful with the interpretation of the tree because there is no support in the node. And the consistency and retention index are about 0.5. So that means that there is still homoplasy, despite the fact that I tried to remove the eventual homoplasy carried by the frontal appendages. Okay, so now if we come back to the main question, are frontal appendages a good phylogenetic proxy? So the idea was to compare my tree with the published tree and see the differences. If the tree was similar, that would mean that it would be a good proxy because despite the modification and the emphasis on the body characters, I still have the same evolutionary signal. But if the trees are different, that should mean that we should be careful with the use of frontal appendages because the signal is different with the body. And in our case, it's more the second hypothesis that is highlighted. The trees are different. They don't have the same relationships anymore. So that's it for today. So I would like to thank first the different structure that helped funding my work. And I also would like to thank in particular the lab in which I worked on because they were really supportive even 2 years after this project. So thank you for your attention and I'm waiting for your questions.